Good morning to all. So in our previous class, we already so our syllabus in this semester, that was immunology. So that was T cell activation and B cell activation and monoclonal antibodies, and the production was completed in our previous class. So those are monoclonal antibodies are mainly helpful. So those are prepared with the help of a single parent cell only. That's why they are considered as a monoclonal antibodies. Actually, so those are prepared by the fusion of myeloma cells with the already immunized splenocytes, spleen cells from the mice. So those two types of cells are going to be fused with the help of a microtiter plate that can take up to 1 to 3 weeks of time. So after completion of such time, so the fusion was leads to cause some different cells are formed. That was a mechanism we were considered as a cloning mechanism. So and these were collected from the single parent cell, single line of cell line was from single parent cell. That's why these antibodies are considered as a monoclonal antibodies. Actually, these were mainly prepared by the, so those are going to be fight against the antigens so which are entered from the outer region of the body, and so with the help of monoclonal antibodies a specific area was going to be so that was they are going to be accommodate to the specific antigen. So the epitope area was also going to be prepared with the help of these monoclonal antibodies on the surface region of B cell. So why? Because these mainly monoclonal antibodies are associated with the B cells. So that's why the B cells that was B antibodies related with the B cell or immunoglobulins. So that was mainly the immunoglobulin area was antibody region immunoglobulin. So that may possess a specific antigen binding site, and a specific epitope region is there. The epitope region is a perfect binding site to the antigen. So that area, on those area was only the antigen was going to be bounded at that area only. So by that reason, these antigens, antibodies were perfectly functioned. So these technology was considered as hybridoma technology. Such type of cells are called as hybridoma cells. And these cells are mainly helpful for the diagnostic use, and also going to be neutralize the specific types of the diseases. Those are cancerous cells. We already discussed it in our previous class. It was the cancerous cells that may having the a number of proteins on the surface of this. So those proteins are going to be locked with the help of these monoclonal antibodies. Then only the cancerous cells are going to be damaged, are going to be neutralized with the help of by the functioning of these monoclonal antibodies. And these monoclonal antibodies are also used so for the diagnostic method. So such type of diagnostic method is nothing but for the after conceiving was occurred inside the woman. So then the conceiving, that was she was either she was conceived or not, that was going to be diagnosed. So with the help of detected with the help of using this monoclonal antibodies. So the strip was available. The strip was prepared by the so with the help of the usage of monoclonal antibodies. The strip may possess monoclonal antibodies. So that was mainly helpful to detect the so pregnancy test was easy today. So by the help of these monoclonal antibodies, but not only the pregnancy test, other diseases are also going to be diagnosed with the help of these monoclonal antibodies. Such type of monoclonal antibodies are mainly, so those are influenza virus and streptococcal pneumonia, that was bacterial disorders, that was asthma and pneumonia like diseases are also going to be identified, detected, and not only those diseases, the sexually transmitted diseases, STDs. So those are only circulated with the help of one person to another person, with the help of only sexually transmitted diseases only at the time of fertilization. So there are those such type of bacteria or so those are transmitted with the help of one person to another person. That's why they are considered as sexually transmitted diseases. So those are herpes and the chlamydia like so diseases or mainly sexually transmitted diseases. Those are two types of diseases diseases are mainly associated with the women, men and women. So those are mainly some diseases, so like the HIV virus like diseases, but they may not be going to be life-threatening diseases, but they are curable diseases. But those such type of diseases are also going to be detected, diagnosed with the help of these monoclonal antibodies. And hepatitis, so that was a viral disease. So those hepatitis B was also going to be diagnosed or detected with the help of these monoclonal antibodies. So these hepatitis was one of the so liver related disease. So it was a jaundice. So was the person was suffered from the liver disorder, that was a jaundice. Means hepatitis. Those hepatitis was also going to be detected with the help of these monoclonal antibodies. But not only these diseases today, and from now from 2020 onwards. From the last year onwards, another disease was also included in these monoclonal antibodies diagnosis method. That was nothing but that was a hot topic for today onwards, till now onwards. That was mainly corona disease. So the corona was going to be, COVID-19 was going to be cured, diagnosed, so it was also going to be somewhat going to be cured with the help of these monoclonal antibodies. So such type of these monoclonal antibodies are prepared, already immunized. It means already immunized is nothing but the person who are suffered first getting the coronavirus, and other person was completely cured from particular the type of disease. That person was considered as a immunized person for corona. So that person may possess a more amount of the antibodies against the coronavirus. So these are those such antibodies are collected from those person. Such type of antibodies or monoclonal antibodies. Those antibodies are may helpful to treat the to cure the corona disease for other persons, mainly more severe persons. So that was the severity was mainly that was so if they are entered into the lungs area, the severity was increased. So but first they are entering into the body, first 2 to 3 days of time they are located mainly in the throat region. So from that onwards they are gradually entered in the lungs area. So by that way, in the traveling mechanism from the throat area to the lungs, so that may considered as the that may so reveals actual severity of the persons and that may also including that may also be so it depends upon the immunity system of the particular person also. So already the person may having a more amount of the immunity, but the virus was not easily entered into the lungs. So but the person who are not having the sufficient immunity for those persons lungs are directly receive the so immediately so less duration of time will taken to entering into the virus into the lungs. That's why so those persons severity was increased on those persons. So that may be life threatening to the persons. Actually, you know very well. I think that most of the persons are died with by the virus corona entering into the lungs and the damaging of the lungs. And not only the lungs, also the remaining the liver and heart, so brain also going to be injured by the effect of these virus. To overcome that problem, to prevent that problem, these monoclonal antibody therapy was also helpful to treat that person, to cure the person from the corona also. So such type of we are discussed in our study class remaining except corona remaining diseases are going to be diagnosed with the help of these monoclonal antibodies, but corona is also going to be cured with the help of these monoclonal antibodies. So nowadays monoclonal antibodies are prepared for fight against about the coronavirus. So those are and monoclonal antibodies are going to be sufficiently fight against for the coronavirus and going to kill that virus. So for binding them, by surrounding them, bind, kill them and digest them. So with the help of these monoclonal antibodies, so that was severe patients are mainly, so most severe persons may given to these type of monoclonal antibodies in the formation of blood plasma. So plasma therapy was a more usage for treating these corona patients. So that plasma may possess that was nothing but that was already immunized person's plasma was more usable for treating the corona to the patients. So that immunized person's plasma was collected and it was given to the person who was currently suffered from the coronavirus. So those are immediately going to be cured with the help of those antibodies related plasma. So that was plasma therapy. Also that may contain the monoclonal antibodies therapy. So these monoclonal antibody therapy was more usable to treat the corona nowadays. So that is about the monoclonal antibodies and their production and their diagnostic use was completed. So till now, our new topic, next topic in this, totally the monoclonal antibodies was from that onwards, the first unit in your syllabus was completed. So from today onwards, we entering into our second unit freshly. So that was mainly about today's our topic was major histocompatibility cells, the structure and functions of today's our topic from second unit. So that was the first topic, structure and functions of major histocompatibility complex MHHC molecules. It was so these are present and second unit, the first topic and today's topic. So major histocompatibility complex cells we already discussed in our previous classes. So why? Because these are associated with the T lymphocytes at the time of T cell activation. We already discussed some amount, something about the major histocompatibility complex cells. So these are major histocompatibility complex cells or the proteinaceous structures which may mainly act to diagnose, mainly to detect the virus, to detect the antigen and neutralize them. So before going to the process, these are the protein molecules, protein substances which may presented mainly these are also called as surface proteins. So and also to the so at the time of surface area, the nuclear, so these are mainly presented at the membranous area, the external membranous area. These may also that was mainly plasma membrane and the nuclear membrane. So these are going to be found at the areas of mainly that was the external layer of the cell, that was the plasma membrane. So these are all that's why these are called as, these are considered as surface proteins. So these are the proteinaceous structures, so which may presented on the surface. These may act so surface region, surface proteins are mainly at the plasma membrane and also the nuclear membrane also. So these may going to be neutralized identify the major histocompatibility complex cells are associated with the T helper cell and T cytotoxic cells. Then they are going to be digest, they are going to be neutralize the harmful substances which are entering into the body. That was from exogenous mechanism, that was entering those harmful substances from the external side of the cell to the internal side of the cell. That was the exogenous mechanism. Or sometimes some DNA damages or occurred inside the cell, and some other the cell was going to be stressed, become stressed. So some other reasons are going to be damaging of the cells or occurred inside the body, natural mechanisms. So by those already damaged cells are going to be die. So that was mainly enhanced, that was going to be increased, the process was going to be occurred, taken over by these major histocompatibility complex cells with the association with the T cells. So the T's are mainly comes under these are major histocompatibility complex cells. So these are cells are mainly 4 types. On those class 1, class 2, class 3 and class 4. So these are the 4 types of major histocompatibility complex cells. So those are so class 1, class 2, class 3, class 4 are there. So only class 1 and class 2 are majorly associated with the T helper cell, T cytotoxic cells. Remaining class 3 and class 4 are mainly helpful for the formation of remaining white blood cells, and also the mature thymocyte cells only, those are presented, but the remaining 1 and 2, class 1 and class 2 are only majorly functioned and associated with the T cells for deactivation of the harmful substances such as antigens. So first we want to discuss about the class 1 MHC molecules. Now class 1 MHC. So you see the diagram for these and functions of MHC. So in these first we want to discuss about the class 1 MHC. First of all, you see the diagram for this. series. So this is the structure belongs to the totally about a class 1 MHC molecules. So it was always associated with the T cytotoxic cell. And so CD plus 8 adhesion molecules. We already discussed it, T cell activation, but also repeated because the topic is totally about the major histocompatibility complex cell structure and functions. On that, so those are mainly 4 types are there: class 1, class 2, class 3, and class 4 are there. So on those, class 1 and class 2 are majorly so always associated with the T cells. On those first is the structure about that was a class 1 MHC molecule series. Totally 3 polypeptide chains are there. That was alpha 1, alpha 2, alpha 3 are comes under the polypeptide chains here. So these 3 are bonded, covalently bonded with each other. So in the inside these sulfide bonds are also there. So covalently bonded with each other. But so another single domain is there that was not bonded with so these alpha 1, alpha 2 and alpha 3 that was variated. That was a beta to M. The beta to M is M means a microglobulin. So this is the total structure about the microglobulin of beta to M. Totally this is about the alpha 1, alpha 2 and alpha 3. So in these two membranes are there, domains are there. So one is mainly membrane distal domain, means it was far away from the plasma membrane. This is the structure of plasma membrane. This is the cytoplasmic tail, means the cell was totally ended. So inside the cell totally the cytoplasm is there. The ending region of cytoplasmic tail, it was considered. The end region of the each and every cell, the plasma membrane is presented. So this is the structure belongs to plasma membrane. That is the why, because these MHC molecules are surface proteins. They are presented mainly nearby the plasma membrane, cell membranes only, nuclear membrane also. So this is the structure of plasma membrane nearby this. So end region of cytoplasm, that's why it is called as cytoplasmic tail. This is the structure of plasma membrane. So from this the domain which was nearby the plasma membrane, that is called as membrane proximal. Proximal means nearby domain. That's why membrane proximal domain. And this is the membrane distal domain, means it was somewhat distance far away from the plasma membrane. That's why that is called as distal, means some long distance. That's why that is a distal domain, and the proximal means a nearby membrane. That is a membrane distal domain, membrane proximal domain, and this area belongs to transmembrane domain. Why? Because it is the area for plasma membrane, and so in these below area cytoplasm is there. It is totally the surface region of plasma membrane. These MHC molecules are located. Why? Because these are the surface proteins. Cell surface proteins means cell surface region is membrane, that is a plasma membrane. So there only these MHC molecules are located. This is structure totally about the class 1 MHC molecules. These are always associated with the T cytotoxic cells. So and another adhesion molecules are there. Those are CD8 adhesion molecules and T cytotoxic cell. Cytotoxic cell means already these class 1 MHC molecules combined with the T cytotoxic cells. So both are going to neutralize, both are going to digest the cell, and that was already damaged cell which was presented inside the body of the organism. So mainly these class 1 MHC and T cytotoxic cell already, so infected cell already, so cell damages were occurred inside the body, or cell was undergoes the stress. So then those reasons are going to be the cell cycle checkpoints are there. Those are CDKs and cyclic dependent kinases, CDKs, so cycling CDK cyclins. So those are the cell cycle checkpoints, and retinoblastoma and protein 53, P53 factors. Those are mainly helpful for cell cycle checkpoints. So if any DNA damages are occurred, then immediately the DNA undergoes a repairing process. So sometimes so the cell was going to be fail to process those DNA damages. So by that way the cell was not usable. So that cell was completely going to be neutralized, destroyed. So that process was mainly occurred with the help of these class 1 MHC molecules only. These class 1 MHC molecules are always associated with the T cytotoxic. Cytotoxic means cyto means cell. Toxic means already that may contain the toxic substances. Such type of cell. So those cells are about the T cell. So those are associated with in the presence of these MHC molecules only, class 1 molecules only. Thus type of cell was going to be neutralized. Such type of cell was going to be degenerated, so destroyed. So that phenomena, so that was also considered as antigens presented inside the body. So why? Because the cytotoxic cell may contains the harmful substances inside the cell. So the type of mechanism, so that was considered endogenous methodology. So inside the cell, already infected cell was going to be degenerated by the activation of association of these MHC1 class 1 molecules. So that type of cell possess antigens inside the cell. So the type of mechanism were performed with the help of these class 1 MHC, and in these alpha 1 and alpha 2, so alpha 3 totally may possess 90 amino acids. 90 amino acids are there totally. So these alpha 1, alpha 2, alpha 3 domains may contain 90 amino acids, up to 90 amino acids. Totally this is about the class 1 MHC molecules, and after that class 2 is there. So totally class 1 was now completed. So it was associated with the class 1 completely. So that is totally about the class 1 molecules, and after completion of the class 2 is there. So that was also same sometime structure. That was some variation is there in the middle of class 1 and class 2. The variation is so these are alpha 1, alpha 2, alpha 3 only combined, but in class 2 alpha 1, alpha 2, alpha 3 and alpha 4 is also located. But in class 1 MHC, so there is no alpha 4. But in class 2 MHC, so there is a alpha 4 is there. So both the 4 domains are bonded with each other. So but that is a variation between these two. So class 1 was completed today class. So class 2 we move on to discuss it in our tomorrow class. So we'll meet tomorrow. Thank you.